Okay, so we have a question from Tamiris. So it is about the voluntary and involuntary muscles. So the usual way to explain it is that voluntary muscles, they are all skeletal, while the ones that we cannot control with our thoughts, these are smooth or cardiac muscle. So the major difference is in the composition of the muscle fibers. So as you can see on the slide, you can see these photos. So this is the skeletal muscle, this is the cardiac, and this is smooth muscles. So if you take, for example, the skeletal muscles, they usually create so-called conglomerate of cells. So they combine their cell membranes, and we kind of have a single huge cell with all these nuclei within the same membrane. This helps them to kind of allocate all these fibers to perform the same single function, so to contract or to relax. And whenever we have the cardiac muscle, so you can see that they are actually are separate cells, and you can define which ones kind of interconnect with each other, and they of course have one single nucleus. And in the smooth cell, we have much smaller cells, and the nucleus in them kind of takes much more space than in cardiac muscles. So this is the main difference between them. Also, I can tell you that whenever we have the contraction of the skeletal muscles, so because let's say that we need to perform a lot of actions, and to do so, we need to spend ATP. And whenever we spend ATP, of course, the inner kind of storage of ATP is just a second, is spent very fast. So if you think about, do you see my screen now?
Yes.
Okay, nice. So you have this contraction. So let's say this is the muscle fiber, and well, we usually have the amount of ATP inside this muscle cell or muscle cell conglomerate. But whenever we run out of this ATP, the actin and myosin, they cannot move along each other. So whenever we don't have ATP, this movement stops, and the fibers, they relax. So actin goes in one direction and myosin goes in another. So to stop this relaxation, very fast our muscles, they turn from the aerobic ATP production into the anaerobic. And if you remember, the anaerobic one is less efficient. So if we take aerobic ones, so this is the respiratory chain, and we have mitochondria involved here. And from one glucose, we obtain about 38 ATP molecules. While in anaerobic one, we get only 2 molecules of ATP, plus a lot of acids. And these acids, they kind of stay in our muscles. So the concentration of acids, it increases. And this gives us the pain. So whenever you feel pain after lots of work, etc., it means that you kind of turn from the aerobic ATP production into the anaerobic one. And then we first need to get rid of these acids, and after that, our pain is gone. okay? And if you take the cardiac muscle or the smooth muscle cells, they usually don't feel this pain because they are very efficient in the production of this aerobic ATP, and they are also having a good bloodstream. So the blood supply is much better in these cells because they kind of in a constant movement, so in a constant function, functioning, let's call it like this. And they kind of allocate all these blood, oxygen, etc., very efficiently so that we don't have this pain, etc. But if we have ceased the blood supply for a second, many of these cells, so this is cardiac and smooth, sorry, smooth. So if we kind of stop this blood supply, many of these cells will die almost instantly. That's why, for example, the ischemia, which means the cease of blood of the cardiac muscle, it is infarctus. And this is very dangerous condition when people need very fast and full medical support, usually in the resuscitation unit. And if you take the muscles of like skeletal muscles, they can survive in these low blood supply. So this is the main difference between them. These smooth muscles, you can find them in arteries and the digestive system, as well as in all the glands, etc. So our glands, so let's say this is a gland, and we have this duct. So first we need to kind of push this secret inside the duct and then push it along this duct. And this everything is supported by these smooth muscles. Can you explain the slide about energy, muscles, myosin, etc.? In this way, so again, you have the muscle fiber, and it is controlled by the nervous system. So the nerves, and of course, it is the voluntary muscle, so it's skeletal. And whenever we think to move this muscle to perform some kind of action, this nerve, it sends this signal. And whenever it sends this signal, so let's make it with this one. So this part, also called the synapse, you can see it here. So this small part is shown here, okay? So this is the cell membrane of muscle. And we have this neuron coming to this muscle. So it kind of touches the muscle. And whenever it receives the signal from the brain, so this is electricity, and this signal kind of releases the special molecules, which are called neurotransmitters. So in this case, we have a transmitter called acetylcholine. And this acetylcholine, it regulates the pathway. So we have special receptors here. And whenever they they are being excited by this acetylcholine. They send the signal to this sarcoplasmic reticulum. And this sarcoplasmic reticulum, or SR, it releases the calcium ions inside it. So the sarcoplasmic reticulum is the depot for calcium ions. And whenever it releases it, so here we have the cytoplasm of the muscle cell, okay? So we have these free ions that are floating everywhere inside this huge cell. And also we have these filaments here. So this filament is myosin. And this is the complex of actin. So let me get rid of these guys. So here we have actin plus tropomyosin plus troponin. So this particular complex, it will look like the usual fiber of actin. And on this fiber, we will have special sites of binding of the myosin. So let's indicate with this one, or okay, so again, the actin filament and the binding sites for myosin. So this is binding site. And because this actin is in a complex with tropomyosin, the tropomyosin, it will kind of block these binding sites for the myosin. So it kind of prevents the binding with the myosin. So again, tropomyosin is kind of antagonist for myosin. And also on this filament of tropomyosin, we will have the troponins. And this guy is troponin. So whenever we have calcium released in the cytoplasm, this calcium will bind to troponin. Okay, so this guy binds to troponin, and together this complex of calcium and troponin, it changes the conformation or 3D structure of the tropomyosin so that it kind of gets away from these binding sites. So it doesn't disappear, it just opens these. So let's make them red now. So it opens these binding sites, and in this case, they are open for interaction with the myosin. So myosin, it has the structure. Again, sorry. Okay, so the myosin has the structure of the filament, and on its end we have so-called head. And this head is actually capable to bind to the binding sites of the binding sites of the actin. So whenever we have the calcium in our cytoplasm, this calcium is binding to troponin, and this troponin changes the 3D structure of tropomyosin and releases these binding sites. So now they are free, and this head, it can bind to this guy. with the help of ATP. So again, ATP comes to myosin. So this is actin. So again, this ATP comes to this head. It hydrolyzes to ADP, so it's adenosine diphosphate plus 1 phosphate, and each of them, they kind of stay here. Okay, so ADP plus P here, and it kind of changes the, not kind of, but it changes the conformation again, so that this head, instead of, so let's show it like this. So whenever we have this ATP binding, this head will extend in this direction. Okay, so towards the next binding site. And after this head extends, it releases this phosphate. And after that, after this release, it will try to get to the previous state like this. So this is the first state, and it will try to get to the previous state. And in this case, it will kind of pull itself towards the next binding site, okay? And that is why we have the movement of myosin along the actin filament. So because of this change of conformation of this head, so it gets to from one binding site to another, and then pulls it towards the other side. And of course, the myosin will go in this direction. And if we think about the whole line of actin myosin, it will also change the distance. So we will have some change in the distance between one side and the other side of this total filament.
And when it like relaxes, it returns to
it just goes back right in the other direction. So we have this calcium going back to this sarcoplasmic reticulum. So our troponin, it loses this binding with the calcium, and in this case, the tropomyosin will return to its inactive state. So to previous 3D conformation, and will close these binding sites of the actin. And because of that, our myosin will not be able to walk along this actin filament, and it will kind of slide back to its previous state. Okay.
Okay, thanks.
You're welcome. Almost the same happens when we run out of ATP. So whenever we don't have ATP, the myosin also slides back, even though we have this calcium in our sarcoplasm. Okay, I hope I answered the question and you all understood it. So next question is from Jean-Saya. Cells with bones, especially about osteoclasts, why do they break down the bone material? Point, why do these cells do it? Okay, so we have a question about the types of cells. And here we have it. So the osteoblasts, these guys are producing the bone tissue. So let's say we have a bone. And let's make it the phalanx. And so the bone tissue here, is made by these osteoblasts and osteocytes. So the osteocytes are just mature osteoblasts. Also, we have these osteoclasts here, and these guys, they constantly remove the tissue or break down the tissue. Why do we need it? We need it for the growing of the blood vessels. So remove bone tissue to give space to blood vessels and nerves. This is the first one. Second one, we will need to remove this bone tissue to kind of recreate the inner structure. So if you take this trabecula, so let's say at some point we had muscles of, let's say 1 kilo of muscles that were binding to this part. So these muscles, they were binding to this part of the bone, okay? And at some point, person started walking a lot or doing physical exercises. And after some time, we have, let's say, 1.5 kilos of muscles. And of course, they didn't change the application of application point or point of fixation to the bone. But since we have much more muscles there, they will pull this bone in either direction, so whatever direction the muscle has. And in this case, the inner structure of the muscle or of the bone is not sufficient anymore to provide stiffness. So it's not stable anymore, if you think about that. And if it doesn't recreate itself in a way to respond to this new load or new functions of the muscles, we might break it because in nature, we always do the stuff that is sufficient for the functioning. But if we had sufficient bone tissue for 1 kilo of muscles, it's not sufficient for 1.5, right? That's why it really needs to kind of remove some part of it and recreate it with much more bone than it was before. So in this case, we remove it, and osteoblasts will recreate it in a way that it responds to the needs of the organism. Okay, so the new muscle most probably will be a little thicker, and the inner structures like trabecula, etc., they will also be recreated because of this new functioning of the muscle. So this is about the osteoclasts and why they do that. It's just because the bone itself, it is the organ of our human body, and it really needs to, let's say that one of these cells died and released a lot of acids, etc., or it has some kind of inflammation inside the bone. So we will need to remove it, and we will need to do it in a very fast way. So these osteoclasts, they will start breaking the stuff to get access to this dead cell and remove everything from this spot. And the other cell, osteoblast, it will get to this place after osteoclast has done the job and recreate the whole region so that we don't lose the stability provided by our bone. Okay, so this is about the different roles of these cells, and the lining cells, again, they are very close to the edge of our bones, and they are needed to provide the communication or relations with other cells and other tissues. And also we need these lining cells whenever we have broken our bone, for example, so that we can start repairing it very fast, okay? So the second question is about the difference between compact and spongy bone. And it is here. So this picture actually shows both compact and spongy tissues. So why do we have the compact bone? It's because we need to have the shape that is constant and stable. We also need to kind of have this protection against the pressure, the heats, and other stuff that can damage our bones. And the thing is, if we kind of fill the whole bone with this compact tissue, of course, we can kind of make these canals, etc., to get our vessels and nerves inside. But in this case, this bone will be super heavy, and it will create a lot of problems because for heavier bones, we will need much bigger and larger muscles. We also, because of these huge muscles, we will need much more blood. Our heart will kind of work much more than it was working before, etc., etc. So just imagine the bodybuilders who started creating these huge amounts of muscles, and they might develop some heart problems or the vessel problems, because our body is not kind of meant to have this much meat, okay? And actually we have it from our ancestors, so dinosaurs, etc. They all were evolving in a way to have the most efficient mass and kind of energy spending. Okay, so we have balance in energy. So this balance means that we spend energy and we obtain energy. So to be able to spend more, we need to obtain more. And whenever we have huge mass of the organism, we of course need to spend energy much more. And this means that we need to obtain much more too. And in this case, to obtain much more energy, we need to again spend much more energy to just get this food, etc. Right? And usually in predators, you spend a lot of energy to catch the prey. And in this case, sometimes we might not get the enough amount of food just to be sure that we actually have enough to kind of spend for ourselves, etc. So to survive. That's why we have these very fine and well-controlled balance in energy consumption and energy spending. And of course, the production of this spongy bone is also a part of this energy balance. So the lighter our bones are, the less muscles we need to kind of lift these bones, but also we need them to be very rigid and stiff. That's why we need this compact tissue on the sides. Second good part about this evolution, that we obtain a lot of space. So inside our bones, we kind of get a lot of free space that we can use for, let's say, storing energy as yellow bone marrow. Or we can use it for protect blood production. Okay, so in this case, you produce blood from these blood progenitors that kind of stay inside the bone. So it is protected against heats, against pressure. Also, this compact bone, it doesn't let a lot of heat fluctuations, which means the kind of stability or balance in temperature inside this part. And of course, our stem cells and the blood stem cells, they are really dependent on the temperature that is in this place of blood production. Let's say that we have this blood production in our skin, and in winter it's very cold outside, in summer it's very hot. So these fluctuations from 40 to -40, they will most probably kill our blood cells or lead to the mutations. And if we have mutations in our stem cells, of course, it's very bad for offspring of this cell. That's why we need the stable and protected place, and usually it is the bone marrow inside our bone. So this spongy bone, it also protects the blood production or producing machine. Also, why we need it? Well, we need this spongy stuff to kind of create the special elements that make our bones much more stable and rigid. So we create this trabecula, and if you look at this trabecula, they are in parallel with usual pressing vector. So let's say our body weight, because here we have the pelvis. So this is femur, or the first bone of the leg. And just imagine the whole weight of the human that gets like this, okay? And if you think about the bones, this vector will be created by pressing from this side and also on this side from muscles. And we need to kind of create the structures that will kind of make the construction much more stable and rigid or protected against these forces. So we usually have them in parallel with these vectors, okay? So these are the functions of spongy bones and the functions of the compact bones.